So this is chapter 24 on amino acids, peptides, and proteins, part 1. Proteins are comprised of alpha amino acids. The alpha amino acids are joined by peptide bonds, and proteins are involved in structure, enzymes, transport, protection, and hormones. Structure of protein has an amino group on one side, a carboxy group on the other side, and it has this alpha carbon of which you have a side chain. As a result, this is called an alpha amino acid. Here are some examples of amino acids, and as you can see, the N and the amino terminus and the C terminus are highlighted in blue and green. And if you react the amino terminus with the C terminus, you form what are called peptide bonds or amide bonds, C double bond O and H, as shown here. Again, NH2 and COOH, that's what amino acids have. Glycine is the simplest amino acid, and instead of an R, it has a hydrogen, as in this case. Amino acids with side chains are chiral, and almost all naturally occurring amino acids have the S configuration. They're called L amino acids because they resemble L- glyceraldehyde, and the optical rotation, positive or minus, must be determined expressly. Stereochemistry of alpha amino acids, as we learned previously, this is an L-alanine in which you have priority 1, 2, 3. It's going in the clockwise direction. However, the hydrogen is coming out at you. As a result, it's an S-alanine. Same thing here with Fischer projections. 1, 2, 3. This is going in the clockwise direction. Again, H is coming out at you. As a result, this is S. You can do the same for the glyceraldehyde and the L-amino acid conformation, and you should be able to do this as per last semester priorities and S and R. Standard amino acids, there are 20 standard amino acids. They differ in side chain characteristics. You can have H or alkyl. Some of them contain OH, some of them contain sulfur, some have non-basic nitrogen, some have COOH, some have basic nitrogen. The essential amino acids are as follows: arginine, threonine, lysine, valine, phenylalanine, tryptophan, methionine, histidine, leucine, and isoleucine. In terms of the amino acids and their side chains, in detail in your book there's table 24-2, which not only gives the amino acids and its properties, it gives also the name of them, the symbol, the single symbol, and the abbreviation, of which you should all know, as well as the structure. Now the structure is not showing the S configuration. However, you now know how to represent it in S, either in Fischer projections or as just regular kind of hashed and wedged bonds. So here are all the aliphatic or non-polar alkyl containing side chains. Things to note here are this has an aromatic group, and here proline is actually connected in a 5-membered ring structure. The ones that bear OHs are serine and threonine, and tyrosine has similar to phenylalanine, an aromatic group, but it's a phenol because it's an OH. Cysteine and methionine have S's in them, or sulfurs in them. Asparagine, glutamine have amides, and then tryptophan has indole. Aspartic acid has COH, glutamic acid COH, and these are the basic amino acids. We're going to talk about isoelectric point in a bit. Complete proteins, they provide all the essential amino acids. They're found in these products. Proteins that are severely deficient in one or more of the essential amino acids are called incomplete proteins, and plant proteins are generally incomplete. So for vegetarians, to get kind of the complete proteins, they should supplement their diets with things like milk. Rare amino acids, so there are modified proline and lysine in the form of 4-hydroxy and 5-hydroxy, 4-hydroxyproline and 5-hydroxylysine, which is found in collagen. D-glutamic acid is found in cell walls of bacteria. D-serine is found in earthworms. Gamma-aminobutyric acid is a neurotransmitter, and beta-alanine is a constituent of the vitamin pantothenic acid. Properties of amino acids: they have high melting points over 200 degrees, more soluble in water than in ether. They have large dipole moments than simple acids or simple amines, and also they're less acidic than most carboxylic acids, as shown here, and less basic than most amines, as shown here. Zwitterionic form, so the major component of amino acids are expressed as the zwitterion, in which the amine is protonated and the carboxy terminus is deprotonated, and so this is what it normally exists as. So this would be the neutral zwitterionic point, which is around pH 6, and then you can go at basic pH, it becomes you have this being neutral and the carboxy being deprotonated, so it would have a negative charge overall. And as you go to acidic pHs, it'll have a positive charge because it'll be protonated, and this would be neutral. Isoelectric point, this is again showing that under acidic conditions, it's going to be protonated and bear a positive charge. Under basic conditions, it's going to be negatively charged, deprotonated, and isoelectric point, it'll be neutral. So the PI, or isoelectric point, is defined as pH at which amino acid exists as a zwitterion, and the PI depends on the structure of the side chain of the amino acid. Isoelectric points, essentially acidic amino acids, have a pH that's around 3, basic pH around 9, neutral around 5 to 6. Thing to notice that at its isoelectric point, an amino acid has a net charge of 0 with NH3 plus and CO minus balancing each other. In more acidic conditions, lower pH, carboxy group becomes protonated and the net charge is positive. Basic conditions, higher pH, amino group loses proton and is net negative charge. Electrophoresis separation. Electrophoresis uses differences in isoelectric points to separate the mixtures of amino acids. So positively charged or cationic amino acids are attracted to the negative electrode or cathode. Negative or anionic amino acids are attracted to positive electrode, which is an anode. And amino acid, as its isoelectric point, has no net charge. So for example, here's an electrophoresis setup, which you have a cathode and an anode and a power supplies. And then here's a streak that contains a combination of neutral, basic, and acidic amino acid. As you can see, the lysine, which is basic, will move to the cathode. The aspartic acid, which is acidic, will move to the anode, and then alanine doesn't move because it's neutral. Reductive amination is a way to make amino acids. You can take the alpha keto acid with excess NH3. This will form the imine, and then you can reduce that imine by hydrogenation with H2 and palladium to generate the amino acid, and this amino acid is racemic. This method for synthesizing amino acid is biomimetic. It mimics the biological The actual biological process involves reductive amination of alpha-ketogluteric acid, the presence of ammonium ion with NADH as a reducing agent, and your enzyme that will catalyze the formation of L-glutamic acid, generating NAD+, and it'll generate just the L enantiomer. Transamination, you can take the amine and actually transfer it to oxaloacetic acid to generate L-aspartic acid and then alpha-ketogluteric acid. This is done by an enzyme called transamination, and this process called transamination is done as such. Amination of alpha halo acid, so you can generate the alpha halo acid via Helfel-Hart-Zelinsky reaction, in which you brominate as a first step and work up with water. This will generate the alpha bromo acid, and then the alpha bromine will be replaced with ammonia by treating, will be replaced by amine by treating it with a large excess of ammonia to generate both the DL and L form. Gabriel malonic ester synthesis, so you can use N-thalidomalonic ester, which has diester. In the presence of brace, it'll deprotonate, and then you'll have SN2 reaction to alkylate. Then in the presence of H3O+, both of them will convert into the acid, and then upon heat, only one of them will release as a CO2. Strecker synthesis is another way of which you can make amino acids, starting with acetaldehyde. To make alanine, you add ammonia, HCN. In the presence of water, this will generate this alpha amino propionitrile, and then upon treatment with acidic water, this will generate the DL-alanine. The Strecker mechanism, first step is protonation, right, with the H+, and then nucleophilic attack, and then forming the imine, of which you can get the mechanism from previous chapter 18. Then it protonates again, and then the CN attacks, quenches the positive charge, forming the alpha aminonitrile. This can be hydrolyzed, which we've talked about previously in chapter 18. Important thing to note is that malonic ester synthesis, you use side chain of the desired amino acid. That must be a good SN2 substrate to alkylate the ester. In the Strecker synthesis, the aldehyde carbon becomes the carbon of the amino acid, so you begin with a side chain, and that has these aldehyde attached to it. So here's a problem. Show how you would use Strecker synthesis to make isoleucine. So isoleucine has a Seq-butyl group, so you want to attach that to the aldehyde, and then you're going to treat it with ammonia HCl H2O. This will generate the nitrile, ammonia nitrile, and then in the presence of H3O+, this will generate the DL-leucine. Resolution of amino acids, so usually only one L enantiomer is biologically active, so you convert amino acid to a salt using a chiral acid or a base, and the result is a mixture of diastereomeric of salts that can be separated by chromatography. That's one way. The other way is to use an enzyme such as acylase that will react with only one enantiomer. And so here's that, where you have both the L and D amino acid. You acylate it, so it acylates the amine terminus, and then you're going to use acylase, and acylase will deacylate only the L enantiomer, and D is unaffected, and then you can separate these two out. Esterification, you can actually protect the carboxy group by esterifying, by adding this benzyl ester onto the group by reacting it with this in the presence of HCl. This will allow for SN2 reaction and will protect it. You can actually deprotect this by using aqueous conditions, and this will regenerate it going back to the proline. You can also acylate the amino group using benzyl chloroformate. In this case, you're having nucleophilic attack. Electrons go up, down, kick off the chlorine to generate the N-benzyl oxycarbonyl leucine. In this case, because it's a leucine that's being protected. And then this can be easily removed as well. Reaction with ninhydrin. Ninhydrin actually is a test of whether you've generated an amino acid. So if you have an amino acid and you reacted with ninhydrin in the presence of pyridine, it'll generate Raman's purple and convert the amino acid into the aldehyde. Peptide bond is an amide bond, as shown here, and it has resonance stabilized, in which electrons from the nitrogen go and form a double bond and kick the electrons from the carbon to the oxygen carbon double bond to the oxygen. And because of this resonance structure, it has a planar it has a planar structure, and because of this double bond by the C and N. Peptide bond, so you can take the carboxy and the amine, and you could do a dehydration reaction to form the amide bond. Polypeptides usually have molecular weights less than 5,000, whereas proteins, which are found in nature, they have really high molecular weights, 6,000 to 40 million. So if we look at hormone, human hormone bradykinin, this is just a peptide sequence comprised of arginine, glycine, phenylalanine, serine, proline, phenylalanine, arginine. As you can see, the N terminus is the amine, and the carboxy terminus is called the C terminus, and they're usually drawn from left to right. Cysteines in the polypeptide chain can also form disulfide bridges. So if you have two cysteines, it can be oxidized to form this disulfide bond, or you can reduce that disulfide bond to the two cysteines. Human oxytocin is a nonapeptide or 9-mer, which has two cysteines, and it forms an intramolecular, that means within the same polypeptide disulfide bond. It links amino acid 1 to 6, and it forms this ring structure. Bovine insulin is comprised of two polypeptides, an A chain and a B chain, that not only have an intramolecular disulfide bond within the A chain, but disulfides bond from the A and B chain, so it has an intermolecular disulfide bond. Peptide structure determination, so one can determine the structure of peptides by cleaving the disulfide linkages, determining the amino acid composition, sequencing from the N terminus or C-terminal residue analysis, as well as partial hydrolysis. And so in the next section, part 2, we'll discuss these as well. So this ends part 1.